My name is Karla Kirkegaard, and I'm a professor at Stanford University School of Medicine. I'm in the Department of Microbiology and Immunology. Actually, I'm moving my lab to Scripps University Research Institute, a new campus in Jupiter, Florida, October 1st. Why am I at this meeting? Actually, it's the first immunology meeting I've been to. And the reason is I was invited because our work with viral susceptibility impacts immunology, and I'm trying to recruit postdocs, and I'm also trying to learn how we can best address this issue. I'll tell you a story about this. So I work on picornaviruses. They're non-enveloped positive strand viruses, also other positive strand viruses, dengue and hepatitis C. But I have a friend, his name is Michel Brahick. He was at the Institut Pasteur for 30 years, I believe, and worked on a virus related to poliovirus called Tyler's virus, which infects mice. And what he showed using classical susceptibility genetics was that there was a locus in B10S mice that allowed clearance of Tyler's virus, whereas in SJLJ mice, they became persistently infected. After years of crosses, it turns out that this SJLJ locus that fails to clear the virus has a dominant phenotype and maps to a certain position on chromosome 10. In that locus on chromosome 10, there was, interestingly, a long non-coding RNA. And I was fascinated by the concept that this long non-coding RNA could be involved in pathogen susceptibility. Now, of course, interferon gamma is there too, so you'd have to be pretty much of a dreamer to think it was the non-coding RNA instead of interferon gamma. And we were told that quite a bit. But in any case, being European, Michel was forced to retire just as they had found this incredible thing. So I had the privilege of having him come to my lab for 4 years, and together with a very talented student named Antonio Gomez, we worked out that it was the long non-coding RNA that confers dominant susceptibility to a virus. Now, how do you confer dominant susceptibility? Usually susceptibility is recessive, that is, you need to have a knockout to be susceptible, but it turns out that this long non-coding RNA is probably anti-inflammatory. That's why even if you have one copy of it, in your mouse, you can't clear the virus. But these mice are more susceptible to inflammation, like sepsis caused by salmonella. So here you have this balanced polymorphism. That is, if you have the RNA, as a mouse, you can't clear viruses, you die of sepsis. Is this RNA polymorphic in the mouse population? Yes. Is it polymorphic in the human population? Yes, the ortholog is. And mutations have correlated with inflammatory diseases such as rheumatoid arthritis and lupus and leprosy, for that matter. So it's very heterogeneous in different human populations, and we're very interested in things like that, balanced polymorphisms, and how they affect why different people are susceptible to different diseases. We're all here, of course, because of what our ancestors didn't die of. So won't it be interesting in this era of personalized medicine to be able to look at particular alleles and long non-coding RNAs and know whether you're going to have a hyper-inflammatory response or not. Anyway, this is kind of a new field for me as a virologist. I'm excited not only about the immunological mechanism, how this long non-coding RNA that controls interferon gamma, pretty much only in CD8 T cells in our system, how it confers reduced inflammation, and how variable it is in human and mouse populations. So that's what I'm here. I'm learning a lot. I enjoyed being part of what was actually a very molecular and very RNA-centered session that I was privileged enough to be invited to speak in. Well, my favorite talk so far was that of Arturo Casadevall. He was invited by the Underrepresented Minority Committee of AAI. And what he talked about was a new synthesis of antibody recognition. And it was very interesting how antibodies that were selected for their high affinity for, their high effectiveness in clearing some pathogens turned out to be catalytic. That is, antibodies don't just work by passive binding. They, in recruiting complement, in one case, it was actually catalytic and cleaved the Cryptococcus cell wall. And he talked about how antibodies interactions are not linearly additive but synergistic. And it was actually quite an eloquent talk and very inspirational to all of us there. Yeah, I'd be delighted to come to AI again. We have a lot of ideas of how to figure out the inflammatory properties of our RNA, also the molecular basis of how a long non-coding RNA can only work at the interferon gamma locus and not other loci. We're interested in the molecular mechanism of that, and I think given the increased role of long non-coding RNAs in immunology, a lot of other people will be interested too.